Hereby, I open this academic ceremony at Maastricht University. My name is Ton de Goey. I am the prorector of the ceremony and also the chair of the degree committee. First of all, I welcome Mrs. Marta Nazzari, and she will defend in public her academic thesis, which is entitled Leveraging Multi-Omics Technologies for Studying the Effects of Endocrine Disrupting Chemicals on Thyroid in Vitro Models. Welcome all members of the degree committee, and in particular the two supervisors who are sitting opposite to me, Professor Dr. F. K. Moore, Associate Professor at the Department of Toxicogenomics, and Assistant Professor Twan van den Beuken, and he is Assistant Professor at the Department of Toxicogenomics as well, both at Maastricht University. I will introduce the 6 opponents later during the ceremony, because if I mention all those names now, you will certainly forget, and I hope not later on. Welcome you all here at the audience in the aula, and I would also like to welcome all followers of the live stream. Mrs. Nazzari, may I invite you to present a summary of your thesis, and I wish you success in the coming hour.
Thank you very much.
The floor is yours.
Okay. Dear Prorector, dear members of the Corona, dear friends, family, and colleagues, both in presence and online. Today I'm going to give you a short presentation of what I did for my PhD thesis that was titled Leveraging Multi-Omics Technologies for Studying the Effects of Endocrine Disrupting Chemicals on Thyroid in Vitro Models. This thesis places itself at the interface of bioinformatics and toxicology. And although the title is quite a mouthful, it contains all the elements that we need to understand what was done in it. And now we will go through them one by one together. First thing, what is a thyroid? The thyroid is an endocrine organ that makes the thyroid hormone. It is located around here in the throat area. And it is shaped more or less like a butterfly. The smallest functional unit of a thyroid is called a follicle. And you can picture it as a ball of cells that is hollow on the inside. On the inside, the thyroid hormone is stored. If we were to slice a thyroid and look at it under a microscope, it would look something like the picture that you can see. So every circle is a follicle with a single layer of cells. And on the inside, the hollow core, which is not empty, but there's the thyroid hormone stored inside. What are in vitro models? It's very simple. Thyroid toxicology aims at studying if and how compounds are toxic for humans. However, we have limited access to the human body as a test subject, so we need to find some proxies for studying it. And we can do so in 2 ways. We can use in vivo models, which means we use animals such as rats, mice, or fish. Or we can use in vitro models, where we grow some cells outside of any organism in a petri dish, and the cells are meant to represent or be as close as possible to the organ that we mean to study. Endocrine disrupting chemicals refers to a very broad category of compounds, mostly of man-made origin, but also there are some also naturally occurring ones that can negatively interfere with the endocrine system. For my project, I studied 16 compounds divided into 4 different classes that can interfere with the thyroid. The first class is called phthalates. They are added to things made of plastic to make them soft and malleable and less brittle. The second category is called PAH, or polycyclic aromatic hydrocarbons, which are generated by the incomplete combustion of organic material. The third class is the organophosphate flame retardants, or OPFR for short, which are added to things, common household products, to make them fireproof. The last is the PCB. polychlorinated biphenyls, which have been mostly used as electric coolants in high voltage capacitors. They have been phased out in the 1970s because of recognized toxicity. However, they are very chemically stable, and even after 50 years, they can still be found contaminating the environment. As you can see, we are exposed to these compounds throughout our daily life. And in general, thyroid disruption can happen at several steps in our body. There can be interference in the thyroid hormone synthesis, so any type of the pituitary hypothalamus thyroid axis, which regulates very tightly the levels of thyroid hormone circulating in our body. But once the thyroid makes the thyroid hormone, it's released in the blood, and there can be interference at any step downstream. So there can be interference of how the thyroid hormone is bound in the blood, how it is excreted by the liver, and any target cell of our body. In fact, the thyroid hormone is very important from in utero development throughout adulthood, and basically all the cells of our body can respond to the presence of the thyroid hormone. What we see in the literature is that when we detect endocrine disruptors in the blood of the general population, so normal people, we often see a decrease in the circulating level of thyroid hormones. However, we do not know if the compounds that I just mentioned in the previous slide can directly interfere with the thyroid itself, so they can have an effect on the organ itself, and how this interference works. The last chunk of the title and also of the thesis concerns omics. What are omics? To describe it in a very simple way, what happens in a cell is that every cell contains DNA. The DNA is used as a template to synthesize at needs RNA. The RNA is then used as a template to be translated into proteins. In the life sciences, the disciplines that study all the DNA, the RNA, and the protein contents of a cell or of a population is called genomics, transcriptomics, and proteomics, respectively, hence the omics suffix. What I did in my thesis, which I think is innovative as I've not seen often done, is use omics for toxicological studies. And in the thesis, in my work, I present 3 different ways that you can do so. So in chapter 3, for example, we took the follicles, which are the pink circles that you can see in the picture. We studied the effect of phthalates on the follicles and the effect that they can have on the DNA and RNA content of the cells. In chapter 4, I studied the effect of all the different EDCs, or the full panel of 16 compounds, and I studied the effect on DNA, on RNA, and proteins. And lastly, in chapter 5, we took the follicles, we dissociated them into single cells, and then we studied the effect on RNA of only 2 compounds, one belonging to the polycyclic aromatic hydrocarbons, and one to the polychlorinated biphenyls. And in addition, we used 2 combinations of sex hormones that mean to mimic the circulating levels of sex hormones that can be found in human male and females of reproductive age, to see if there can be different hormonal contexts can affect how the cells can respond to endocrine disrupting chemicals. One of the beauties, but also the challenges of bioinformatics, is that you generate a lot of data. And just to give you an idea what that means, every cell in our body, the human genome is around 3 gigabases long. Multiplied by the 18 compounds that I use for chapter 3, it means having information about 54 gigabases, which is 54 billion bases. Every time that we would study an RNA sample, we would detect around 50,000 molecules. Multiplied by the 408 samples that were generated for this thesis, it means having information about 20 million molecules. Same for proteomics. Every proteomic sample had around information of around 10,000 proteins. which means having information about 870,000 proteins. But this is still not enough, because sometimes numbers are hard to picture. What does that mean? So if we were to print all the DNA information that we generated, we would fill 18 bookshelves from floor to ceiling. If every RNA molecule that we detected was a ball in a ball pit, it would fill up a cube of 20 meters. That is almost a 7-story building. And why am I telling you all this? Well, because when you have so much data, you don't have enough time in your lifetime to look at all of it, and you have maybe more fun things to do. So what is necessary in this case, if you want to gain some insight that can be useful for you, you need to look at the data through the lens of a biologist to try to gather some information for your purpose. And that is what I try to do in my thesis. And I will show you in the next slides just a small taste of what that means, and what I think are some interesting findings for thyroid toxicology, which was ultimately the aim of the thesis. So for example, in chapter 3, where we studied the effect of phthalates, we saw, for example, that all the 4 phthalates that we had in our panel would upregulate the fatty acid metabolism, or they would all downregulate important signaling pathways or the extracellular matrix organization. If you think that the follicle is a 3D ball, maybe interfering with its structure can also mean compromising its structural integrity and then its proper functioning. In chapter 4, we saw that the thyroid can react to polycyclic aromatic hydrocarbons by upregulating some detoxifying enzymes called cytochromes. And that is something that I have not seen described in thyroid models before, except for some thyroid cancer cell lines. And also the potency of these compounds, there were 4 in this class, is coherent with what is described in the literature. What does that mean? That even if the thyroid is not a detoxifying organ, like for example the liver, it can still react to the presence of toxicants in a way that could be deleterious, because these compounds are converted by detoxifying enzymes, but funnily enough, toxic compounds are made that are carcinogenic for the cell. In chapter 5, we saw that the same level of these detoxifying enzymes are more elevated when you have the female mixture, so you have a combination of sex hormones that is representative of what you find in human female of reproductive age. Conversely, when you have a male mixture, you see that an upregulation of genes involved in inflammation. So maybe there are different hormonal contexts can affect different ways that the thyroid can react. So they highlight a different sensitivity of the thyroid to the presence of toxic compounds. So these are some of the results that I think we found that are interesting for thyroid toxicology. And if we want to just have some take-home messages from the thesis itself, so why is it relevant? Why is it important? I can at least give 4 reasons why. One is I showcase some ways in which you can use omics for toxicological studies. We observed some alteration in pathways, gene expression, microRNA expression, and proteins that I think provide some interesting hypotheses to be further pursued. We tested how the follicles that I kept mentioning can perform to be used for toxicological studies. In fact, these follicles were developed by our collaborators in Brussels, but not for toxicological purposes, not to be applied necessarily to toxicology experiments. And lastly, we generated a wealth of data on thyroid in vitro model exposed to endocrine disruptors. When I started my PhD, I could only find one data set in public available repositories that was derived from an experiment studying the thyroid exposed to endocrine disruptors. And with this, I think we really gave thyroid toxicology the attention that it needs, and I think it is lacking compared to more studied organs such as the liver or the intestine. With this, I want to thank you all for your attention, and I give back the word to the prorector. Thank you.
Thank you very much, Mrs. Nazzari, for a clear summary of your thesis. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Marta Nazzari. He is Professor of Analytics in Systems Imaging at Maastricht University. Professor Nazzari.
Thank you, Mr. Prorector. Dear candidate, first of all, congratulations with this in-depth bioinformatics, but also molecular research and shining light on what's going on. And also for the promotion committee, I think it's very solid work in finding its way into toxicology and the mechanisms thereof. But as always, there's questions. And I would like to ask one of the paronyms to read proposition number 7 or bullet point 7. Or better, that's the third bullet point of this second group.
Proposition number 7, transcriptomics is the Pandora's box of hypothesis generation.
Yeah, that's a very interesting one because Pandora's box opens all human evil. So what's the human evil here? Transcriptomics or hypothesis generation? And then we have, I hope you choose the right one, otherwise we have a heavy discussion here.
Highly esteemed opponent, thank you for your compliments and your question. When Pandora opened the box, there was not only evil inside, but also hope. This is the version of the legend that I know. So it is true that it's one of the challenges also that I think I faced, and also a bit of what I think I would have liked to do more. So there can be I think I found a lot of interesting hypotheses, but I would have liked to have experimental validation to see if those hypotheses were actually true. So
it's much more you worry that you don't get really data which are not validated, or is there hope that sometime in the next future, nearby future, people start to understand what's really going on? What's the problem here?
I think we got some really interesting hypotheses, and what also I think it's in general for all fields of study. Knowledge is compound, right? So maybe what we published on chapter 3, for example, with the phthalates, we describe how phthalates can interfere with the fatty acid metabolism, and somebody else will pick up on that and will be able to prove or disprove.
So still hypothesis is there, and that's what your hope is that will help.
And it's valid
that's the good answer. Yeah, that's the good answer. Otherwise, I have to give my title back to the university or something like that. But let's discuss that, because actually you opened a little bit a box of Pandora by doing your research and telling, hey, there's a lot of strange things going on, and here's maybe an explanation. And surprisingly, and also in your introduction, I was a little bit shocked, because actually the authorities claim that you should not be accountable for understanding the mechanisms. You just give the compound and you look if it's toxic or not, and who cares? And it's a little bit strange. It's written in your introduction. I forgot the page exactly. And on the other hand, increasingly the authorities ask you to have a rational design of safety for molecules. So to your opinion, do we need to go into molecular mechanisms to really understand toxicology?
I think it's the toxicological risk assessment, so regulators that just need an information of is thyroid hormone, for example, in my case, thyroid hormone production altered or not? Do you see changes in thyroid hormone levels? That's for toxicological risk assessment. But also, I think you need to understand what are the compounds doing inside your cells. Like, how are they disrupting? What is the mechanism that is being disrupted? So, mechanistic insight, I think, of course it's important. Otherwise, how can you know?
Yeah, okay, I completely agree, because the pharmaceutical industry is held to understand the mechanism and the rest of the world should not. They just give and look if it's toxic. Anyway, I think the basic philosophy and hypothesis is that even water is toxic, so all molecules are toxic per definition. Now, I would like to go a little bit into the molecules, and you said, well, shining light on the mechanism. As for biologists, I would say hardcore chemists are even more important. But if I look to the structures of the molecules you used in your testing, I have a lot of questions there, especially the PCBs. You chose there one coplanar, the 126, but 77 and 81 are very well-known, very strong toxic coplanar. So why did you choose all these molecules in your thesis?
Yeah, of course. I have to be honest, so my project was part of a European consortium in which there was an endocrinologist. And I started my PhD 6 months after the project started. So it was not up to me to choose the classes and the compounds. So they were chosen beforehand by somebody else in the project.
Yeah, it's a little bit, yeah. But next time, please ask them why and disagree with them, because a good scientist will disagree per definition. Yeah. But okay, then I understand where it's coming from. But especially linked to page 161 and looking to the polycyclic aromats, only BKF, and I really do not understand, activates in your cell systems the CYP1A1 pathway, yes. And the others are not. And my question twofold is, do you really understand why only this aromat is doing that from a structure point of view? And then activating the ZIP system, is that not activating a detoxification process of the compound itself? So that's good.
So to answer, first I will start from the second question. So once polycyclic aromatic hydrocarbons are detoxified by the cytochromes, they are converted into carcinogenic compounds. So for example, BAP or BKF, they wouldn't normally be as themselves toxic, but because of they are used as a substrate by the cytochromes, they become carcinogenic. So that's the way that it works in the. That's how the cells work.
So actually we are transforming a molecule to get rid of, so it's a hydroxylase, so you want to get rid of it, and suddenly they become even more toxic.
Yeah, exactly. Because
a little bit of prodrug idea.
Yeah, exactly. Because normally, hydrophobic compounds are converted in the body to make them excretable into polar compounds, right? But this is this transformation that makes them toxic, in this case of the polycyclic aromatic hydrocarbons. Okay. And to answer your first question, the potency of the 4 compounds, meaning how intensely they can increase the expression of the cytochromes, is different for example, for these 4. In general, polycyclic aromatic hydrocarbons have different potencies. BKF, among the 4, is the most potent, meaning that it can induce the expression of the cytochromes more efficiently, let's say.
I think we will have a lot of debate, maybe later on. I want to give a question back to the prorector. Thank you.
Thank you, Professor Honing. The next opponent is Professor Juliette Lechler. She is Professor of Toxicology. She is not here, but she is present online, as you see on screen. And she is associated with Utrecht University. Professor Lechler.
I'm unmuted now. Good morning, everyone, and thank you very much for this opportunity to attend to this excellent PhD opposition, and my congratulations to the candidate with her thesis. I enjoyed reading it very much. and my compliments are extended to your supervisory team. So dear candidate, well, I enjoyed the previous discussion very much, and I feel like I have to defend myself as a toxicologist. We are certainly very much interested in mechanisms, and I must say you selected a very general definition for the class of chemicals called endocrine disrupting chemicals in your thesis. The European definition of an endocrine disrupting chemical is a chemical that has an endocrine mode of action or mechanism, as well as a chemical that causes an adverse effect through this endocrine mode of action. So a definition about something that negatively interferes with the endocrine system is a little bit too limited. We certainly are looking into what is that endocrine mode of action. So my first question to you would be, what do you consider an endocrine mode of action in the thyroid?
Highly esteemed opponent, thank you for your compliments and your question. It's true that it was a simple definition to make it very understandable. That I agree. Interfere with the endocrine system. It's true that if a compound is just carcinogenic and it affects the thyroid, then it affects the production of the thyroid hormone, but not at an endocrine level. Let's say it just disrupts the cells, and then the organ is compromised. So there are, in the thesis, I include some of the key characteristics of what the NIEHS defines as endocrine acting, and I define an endocrine disruptor as something that can interfere with the the hormone production, but at an hormone level. So for example, if you interfere with how the hormone binds to the blood, then it's an endocrine action. If you interfere with the protein that synthesizes, for example, TPO, the thyroid peroxidase, which is deputed to synthesizing the thyroid hormone, then that is an endocrine disruptor. So anything that has a specific effect on the for example, in the thyroid, you ask for the thyroid with the thyroid hormone production. So any of the key proteins or thyroid markers inside the thyroid, so TPO or duoxa or duox1 and 2, all the enzymes that are taking part into synthesis of thyroid hormone, or also proteins that are deputed to interacting with the thyroid hormone itself. So transthyretin in the blood, for example, or the iodinases in the target organs.
I'm going to interrupt you there. I'm going to interrupt you there because I really want to focus on the thyroid. So you've mentioned some specific endocrine-related genes and targets in the thyroid that can be disrupted by chemicals. So which of the chemicals that you studied do you think has the most evidence for, or the class of chemicals that have the most evidence for really affecting these endocrine pathways in the thyroid?
The problem is that there isn't much evidence of interference with specific proteins. So I would say of the 4 classes, because we use also known thyroid interfering molecules like methimazole and sodium perchlorate, but those are not in the 4 classes of EDC. I think for what there is most evidence are PCBs. PCBs can have been shown to cause hypertrophy of the thyroid, interfere with the TPO, interfere with TSHR signaling. So if I had to pick, PCBs would be the candidates in this case.
Okay, and I appreciate, always the first thing that comes in my mind, PCBs have been banned for a long time, but of course we still measure them in human blood, so we know they may be banned, but they're still out there. So what I'm interested then is to know, knowing that PCBs work so specifically on the thyroid and endocrine mechanisms in the thyroid, could you identify a panel of genes that could really help us to pick up these fingerprints or pick up these mechanisms of other chemicals? So maybe that's my first question. You talk a few times about OECD testing, that it could use a panel of genes. What are the genes that you think should be measured in vitro assays or even in animal assays that could really help us better understand mechanisms?
Yeah, so I would say all the genes that are involved in the thyroid hormone synthesis pathway, so the TSHR, the TPO, duoxa 1 and 2, the iodinase 1, so all the genes that are actively having a role in the synthesis of the thyroid hormone. But I also think, take for example, I think we're still lacking some information on more general, let's say, pathways. For example, in chapter 3, right? I mentioned we found downregulation of pathways related to TGF beta signaling and other raw GTPases and so on. And maybe those have also an important role in the proper functioning of the cell for synthesis of thyroid hormone. So the first that come to mind are the genes directly involved specifically in the synthesis of the thyroid hormone. But maybe there are other cells, other enzymes or genes coding for enzymes that can be that are more generic because they are active also in other cells, but maybe the thyroid is particularly sensitive, or they have an important role in the synthesis of the thyroid hormone that can be interesting to look at. It can be important to look at.
I was along those lines. I was really interested in figure 1 in your discussion, where you let's see, it's somewhere on page 254, where you say, okay, there was only one data set from the thyroid, but there are hundreds of data sets from these chemicals on other tissues. Have you tried to find, common genes or these candidate genes that you think could be really important for determining thyroid disrupting chemicals? Have you looked across those different target tissues to try to identify some of these common sets of genes?
You may give a short answer, please.
Okay, it will be very brief. So throughout all the 4 years, I would read a lot of literature of just PCB and PAH in some other organ in the liver. It's upregulating some UGT, I don't remember the spelling out, but UGT.
Yeah,
and
UGTPs.
Yes, but they would not be altered in my samples, at least, in what I was looking at. So I looked at other genes that were found as being disrupted by these 4 classes, and I couldn't find any correspondence in my samples, at least.
Okay. Thank you. That would have been a promising pathway. Thank you so much for your questions and your answers.
Thank you, Professor Lechler. The next opponent is Professor Frans Ramakers. He is Professor of Molecular Cell Biology at Maastricht University. Professor Ramakers.
Yeah, thank you, Mr. Prorector. Dear candidate, lots of congratulations with this beautiful book. A lot of data. Also, congrats to the promotion team, of course. In view of the time, I would like to immediately go to my question about how representative the model system is for the human thyroid. You are using transfected mouse cells, which are grown initially in a medium that contains bovine hormones. And then you use what is called a supplemented differentiation medium to get what you hope, a thyroid-like structure. Mm-hmm. But you got me really worried when I read on page 146 of note that most thyroid markers are inconsistently or not detected in these bodies, the thyroid bodies. Yeah. So how representative are they?
Okay, so highly esteemed opponent, thank you for your compliments and your questions. I will start again from the last remark. The markers that cannot be identified are from proteomics, and that is not that they are not expressed, because, as I said, these follicles have been first developed by our collaborators in Brussels. So they have made a lot of immunohistochemistry. They see the proteins using antibodies, so they see that the protein is there. I think this is a limit of detection of the proteomics. I had no control over the proteomics data sets being generated because they were generated by our other collaborators in Ireland. So, but when you look at the RNA, we can always find them. So the proteins and the RNAs are there, just that with this data set that were generated with the proteomics, they were not detected. Also,
so can I interrupt you? So what do
course.
you find? Do you find hormones, or do you find, let's say, what is called the PAX8 or NX2.1? Those are proteins that are apparently used to differentiate, to get these cells differentiating into the thyroid cells, but you introduce them into the model.
Yes.
And so it's logical that you detect them at the mRNA level, apparently not at the protein level. So they are not expressed at the protein level. But I was wondering, have you seen any hormone production in these organoids?
So again, I was not responsible for developing the model, so I have limited knowledge from what I learned over these 4 years. It's true that the expression of PAX8 and NKX2.1 is forced at the beginning because they are transfected, but then I've been told by the people that developed this model that they see a switch from the expression of the exogenous to the endogenous copies of these genes. So the follicles, when they are mature, which is the stage of the model that we use, they are expressing the endogenous version of the genes. And also there is T4 production, T4 and T3 production, and they could see this via immunohistochemistry with the antibodies for the hormones.
Sorry, I've read the original paper that described it's quite old, and then they look at, with immunohistochemistry, at those proteins, a nuclear protein and a cell adhesion molecule. Yes. And actually, those are the only molecules that they look at. Okay, so we have to believe that the system that you use, because you have not looked in the microscope to these organoids yourself. You obtain them from another group, I understand, and then do the studies that you're good at.
Sometimes I look at them as well, but just under a bright field microscope. So I could see that they would look like similar. Yeah, I did. Yeah, I had a bit of a wet lab part in my PhD, not so much, but.
If I have 1 minute left, Mr. Prorector? A little bit. I was wondering, how reproducible are your data? Because, as I mentioned before, you're using fetal calf serum, for example, to grow them initially. We know that fetal calf serum has different levels. If you use different lots of fetal calf serum, it's just an example because you use a lot of reagents to differentiate it. But if you use different lots of fetal calf serum, you have different levels of hormones, female hormones. So how reproducible are the data? Do you compare the model systems that you produce every time during your experimentation? And do you detect? Do you have an idea about the reproducibility of the data?
So I don't have to defend this model because I didn't develop it myself, so I can be critical about it. That is something that we have seen. I think it can be reproducible in terms of can we study the expression of thyroid genes, because they are always there, and there are thyroid follicles. It's embryonic stem cell derived, so there are the other types of cells which are not thyroid that are that just develop during the differentiation process. And something that I think is a limitation of what I had at hand is that the relative percentage of thyroid follicles and other cell types is not consistent. So, and the only way I think to make this reproducible is to use techniques that allow you only to then select thyroid cells. For example, via flow cytometry, or with single cell, which is what we did in chapter 5. So we can really see what the thyroid is doing. So I think that is a limitation of the model.
Thank you. At least you thought about it. That's, that's
I
very important.
thought about it a lot.
Thanks a lot.
I had to think about it quite a lot.
I would like to give the word back to the poet.
Thank you, Professor Ramakers. The next opponent is Professor Theo de Kok. He is Professor of Population-Based Toxicogenomics at Maastricht University, and he is also Secretary of this degree committee. Professor de Kok.
Thank you. Dear candidates, congratulations on this achievement, because an achievement it is also for your supervisors. The thesis looks really nice. It's a hardcover. You really see that really seldom, and it almost feels like an insult to write in it, but then again, that's what it's meant for, and it means that it's read also very carefully. I would like to immediately go to chapter 3, if that's okay for you, on page 97, figure 4, which is rather complex, I would say, colorful and extremely tiny. So I had to use my own tools in order to read what it is. And if I understood it correctly, you use Maastricht Pro as an art tool to analyze those relationships and look at gene expression patterns. The clusters are not comparable for the different compounds because they may have different shapes. The colors are for the different exposures, and the y-axis reflects expression values without having any unit. You may comment on that later. My question is, because there are so many different shapes, for instance, for the black ones, DEHP, cluster 1 and cluster 5, or the fourth one, which is DNase 1, why are they not combined? Because they have really the same pattern.
Why are they not? Sorry?
Yeah, if you these are all these hill-shaped curves. But the clusters are really small. They have two. So my question is, it depends probably on how the defaults are set, but wouldn't it make more sense to have larger clusters and then integrate that in these measurements?
Yeah, so Ali, esteemed opponent, thank you for your compliments and your question. I think so. So I use Maastricht Pro, and as you say, in the settings, you can decide how many clusters you want to make. It's from 1 to 9. I think that, for example, cluster 7 and 8 were separated. I mean, they have a similar shape, but they are two different clusters. And I think that is because of the average level of expression. So cluster 7, for example, is 200 to 800 normalized read count. The rest is
that was the other question. It's normalized read count.
It's normalized
because
read
it doesn't
count.
say that. Okay.
Oh, okay. So I think both, because when you have a similar shape but different clusters, it's because of the average base count difference.
Okay.
And it's true that I could, have fewer clusters, and then try to optimize the amount of curves that would be generated for every compound. That is true. That could be done.
Okay. So I was wondering why you then choose to have so many different clusters that looks quite similarly actually.
Yes.
And then you say that at higher doses are consistently different from the rest, which makes them suitable to establish a point of departure. And I see also many U-shaped curves, like for DIDP, which is in red, cluster 4, but there's many other ones. And in my opinion, that demonstrates that it's not consistently different, because the highest dose actually resembles the zero exposure. And that does not give you, at the highest dose, the point of departure. Can you explain that?
Yes. So I think that the point of departure can be relevant for other genes. For example, CALF6 in the IMP, which is green, or cluster 3. So you really see like the highest dose really spikes up.
Yeah, but you say it's consistently. That means overall all genes, I guess.
Yes,
so that the gene indicate that there is a point of departure at the highest dose, but in my opinion, it may also be somewhere in the middle, where you see a a different response than at the higher dose, which may be at the low level if it's bell-shaped or at the highest.
That is true. So yeah, I agree. So if, to be more precise, the point of departure for the highest dose should be for a subset of genes. But it's also true that the highest dose is always consistently different from, or always, most of the times, consistently different from the previous doses.
Yeah, but not from zero. So
not from zero, and that is something that we see with endocrine disruptors, that they have non-monotonic dose response curves.
Yeah, you mentioned that several times, but I'm satisfied with your answer. And then at page 98, that's the next page, you exclude the highest dose for determining the differentially expressed genes.
Yes.
And why would you include the most interesting dose? Because that may be the most relevant. That's in order to establish the differentially expressed genes as compared to no exposure.
Yes. So we were looking for genes that were consistently up or down regulated. So if we wanted to, considering the previous picture where often you see the highest dose is either similar to the zero or similar or just the highest, so where you can see the point of departure, the expression would be discordant. Basically, all the first four doses would be very low and the fifth would be very high. So when you do differential expression analysis, you wouldn't see differential expression in the sense of upregulation or downregulation of gene. If you wanted to do differential expression analysis considering the highest dose, I would put it separate from the other doses.
Okay, I would like to, in view of time, move to chapter 5, in which you describe on page 157 the random forest model that you use to do class prediction for EDC. And then you have a very high accuracy of the prediction. That's in table 3. Now, how does that work? So that's quite impressive, but you don't describe how you actually, how do you actually measure that? So do you make a discrimination in a test set and a validation set? Because otherwise you use the same data to predict what you trained the model on.
Can you please mention
that? That is on page 157.
And you may give a short answer, please.
Okay, it's 157. Oh, okay. I was looking at the picture and I wasn't understanding. Okay, it's just for the random forest. So no, we split the data into 75% of the data is training, and the rest, 25, is a test.
Okay, that is very reassuring because it's nowhere written. There isn't even a method section on how you did the random forest. And so if you didn't do that, then these accuracies would actually mean nothing.
Yes.
Okay, that is. I'm very happy with that answer. And then I give the word back to the prorector.
So that is solved.
Yeah, then it's solved.
Thank you, Professor de Kok. The next opponent is Dr. Steinboes-Koort. She is Associate Professor at the Department of Bioinformatics at Maastricht University.
Thank you. Yes, dear candidate, it was a pleasure to read your thesis, and it also gave me some insights in how endocrine disruptive chemicals can affect molecular changes in thyroid models. And I also want to extend my congratulations to your supervisory team. And as you already mentioned in your introduction, you generated a wealth of data. And although I think you already explored it in depth, there are still some hidden mechanisms there. So I think there's still much more to get out of all the interesting data that you obtained. And also there may be more insights and mechanisms. So not only hypotheses that come out of it, but also some more in-depth. I would like to look at you at chapter 2. Where you designed a workflow for combining microRNA and mRNA sequencing so that you can simultaneously measure that. And you designed your own workflow. I think that was impressive there. But to see how it worked, because there was none available, and that's also one of the propositions that is dedicated to that. But there was already one that was applied but that wasn't suitable. And you compared that one, and that was acceptor PT. And then in table 3 of page 48, you mention all the differences between the two approaches. And one of them is that the ensemble version is different. So it's outdated in the existing package. Yeah. And I wonder, how does that affect the comparison between the two pipelines? Could you elaborate on that?
Yes. So, esteemed opponent, thank you for your compliments and your question. Indeed, what are the effects of different genome builds? Mm-hmm. So every time a different genome build is presented, there will be differences like into how the genes are described, right? The position and how long they are, the introns and so on. I think I think that for protein coding genes, maybe it doesn't affect so much the results, but more for the small RNAs. Mm-hmm. So there are a lot, both, like one of the purposes of EXERP was initially designed to study small RNA, circulating small RNA populations. So you really need to have, I think, an up-to-date version as much as possible if you want to do that. So I think it doesn't impact so much the expression of protein coding genes as much as it impacts the short RNAs and all the different subsets of RNAs that have been annotated.
Yeah. Okay, thank you. And if I then look further in the chapter, you indeed look at also differentially expressed genes and differentially expressed microRNAs. And if you then compare the two methods, then you see although the amount of expressed genes and microRNAs is similar, they're not completely overlapping. And then based on these differentially expressed, you look at pathway analysis, and then if you compare the outcome of the pathway analysis, you see that your method is more sensitive than when using the other pipeline. Could you explain that? Is that then something that you would have expected based on the amount of differentially expressed genes and the amount of genes that you could map, depending on the two pipelines?
The problem of EXERP, the problem of EXERP is that the way it was designed, it wasn't very compatible with ComboSeq, which is the type of library that we used. Mm-hmm. In fact, from ComboSeq, you get fragments of protein contingent, so poly-A species, and those fragments would often be longer than basically, you cannot have a complete read that ends with the 3' adapter that EXERP expects, so it expects fixed 3' and 5' adapters. So this is all to say that a lot of maybe the expression of protein coding genes would be down-estimated, underestimated. And so it makes sense that there would be more, like the read count of genes would be higher for CODA. Yeah. And reflect more the real levels of expression in our samples. And then this would mean that you could have more differentially expressed genes, and hence the pathway analysis would maybe detect more pathways, just because there are more genes that you can use for your analysis.
Yeah, so that again then shows the strength of your approach compared to the other one. And now, so you have this really nice method to simultaneously measure microRNA and mRNA. You look at the changed genes, but I don't see that you integrate the two types of measurements. Do you have any idea of how you could do that? How you could link your changes that you see at microRNAs with your mRNA expression?
I tried to do that. So I would, for example, look at targets. Actually, so targets of microRNAs which can bind to the mRNA. Yeah. So microRNA, mRNA targets. I would do that in with Cytoscape, maybe. But
Cytoscapes and network analysis programs, but you still need some information about the targets, right?
Yes.
So where did you get that?
I got that from MIR-base and
MIR-base.
MIR-tar-base, where you can have the interactions or like putative genes where your microRNA can bind.
Yeah, so you have some prediction algorithms that could predict whether indeed a microRNA is a target. I think that you would really benefit from also looking at those interactions. And thank you for answering my questions. I give the word back to the prorector.
Thank you, Dr. Stein-Bouscourt. The next and also final opponent is Dr. Gieselbrecht. He is Associate Professor at Merlin Institute for Technology-Inspired Regenerative Medicine at Maastricht University. Dr. Gieselbrecht.
Thank you, Mr. Prorector. First of all, I would like to congratulate you on this excellent piece of work. So I really enjoyed reading it. It's a really nice thesis. I also like the design a lot. I would also like to extend my compliments to the supervisors, who obviously did also an excellent job too. I do have a few questions. I would like to follow up a bit on the questions of my colleague here to the left about the in vitro models and kind of the relevance of the in vitro models that was used in this project. And you also kind of mentioned that in your presentation, that it sometimes was a bit difficult kind of to get really kind of biological insights. You get a lot of data, but then you would like to see kind of also kind of methods and tools where you can basically also validate these results. So based on kind of, well, one of the goals that I understood was also kind of to look at low doses, right, of for example, endocrine disruptors in this project. So looking back at this project and from a data analysis point of view, do you think actually the approach in the project was probably not a good one, kind of to combine a very complex in vitro model with a very innovative and new analytical tools and methods? So looking hindsight, kind of, would you change this concept, or for a future project, would you kind of rather choose a different design for such a project?
Esteemed opponent, thank you for your compliments and your question. Yes, the short answer is yes. So I think that I still have my reservations for the results that I got from bulk analysis, because it's a mixed population. So the chapter that I have almost no reservation is chapter 5, where we use single cell. So either in hindsight, if I could just decide what to do, I would use either an in vitro system that is a pure population of cells, so maybe organoids derived from patient material, that is something that has been described, or still growing primary cells and testing those, but then you have just follicles, and everything that you see is a reaction of the follicles. Or if you were to use a mixed population, then you need to use something that allows you to dissect the different responses. So single cell, or with our collaborators, we sorted the cells to sort the GFP positive, which are the thyrocytes. So I think the greatest problem, if it wasn't an omics work, so it was just measuring just thyroid-specific genes with qPCR or immunohistochemistry, then this would be perfectly fine. These cells would be perfect. This would be very good. But
maybe can I interrupt you? And maybe because you mentioned kind of also the cell sorting and the single cell RNA-seq, and I also saw that in your thesis that you kind of highlighted that kind of as a possibility, kind of to kind of ignore kind of the rest of the cells within the in vitro system, and there are for sure a lot of cells which are kind of have more kind of lung background or something like that, or other tissues. But then I'm still wondering, well, you can maybe sort those cells or look at a single cell level at more the thyrocyte-like cells, but in the end you still have the same cells in your in vitro systems, and those cells of course communicate with each other. So I'm not sure, so how do you see that? So do you think that might be kind of just solve the issue, so we just kind of sort the cells that we're interested in, or do you think, well, eventually also, again, still the in vitro model needs to be optimized in that respect?
I think that, as you say, cells communicate, so maybe the thyrocytes that we have in our system communicate with the lung cells. But that is not really, I think, physiologically relevant because the thyroid is an organ, its own organ, and doesn't communicate with the lung. So the cell-to-cell communication, in this case, maybe we would find some results, and they could be interesting, but I don't think they would be very physiologically relevant. So if we see the way that our samples were produced, right? The cells were exposed for 5 days, for example, or 4 days. Then they were dissociated and analyzed straight away. So the effects of cell-to-cell communication with other types of cells would be contained in the cells, in the RNA of the cells, if you're fast enough to process them and the RNA doesn't degrade. And then you would see it with sorting. But otherwise, any information of cell-to-cell communication, I wonder about the physiological relevance that you would find. That is something that we mean to do, not me, but my supervisor and people that will handle my data in the future, look at cell-to-cell communication, what are the other cell types doing. We have had discussion on the relevance of doing so, physiological relevance of doing so.
And yet you kind of also introduced then another level of complexity by kind of treating the cells with kind of different kind of hormone cocktails, right? Kind of to mimic a bit the situation in male and female individuals. And you do see differences kind of in the response in your data. But I was wondering, well, what kind of cells were used kind of in the project? Were these kind of, did they have kind of male or female background, genetic background? And well, if you treat kind of male cells with female hormones, is this not then creating, for example, a conflicting situation for the cell? And is that maybe the result why you basically see differences in your male and female treatment? Could you maybe comment on that?
Yeah, so the cell line, the embryonic stem cell line that was used to differentiate the follicles is a female one. So using this kind of combination of hormones for real organisms is just an approximation that I understand. So could be some articles.
Yeah, Mr. Nazzari, I have to interrupt you. The time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and in particular the quality of your defense. And please await our return here.
Thank you. Thanks.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that.
Now
Okay,
thank you.
I can't read your mind. Years can't fly by now. Don't waste all your time. Cause I'll go the extra mile. Long road to the south side. 7 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. all in, it's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you grow. Outside, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side. 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and not.
Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Mrs. Marta Nazzari. The degree committee has assessed the quality of your thesis and the quality of your defense, and in view of its positive verdict, the committee has granted you the degree of doctor, also taking into account your previous qualifications. Professor Clément is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. And I invite now your supervisor to take the floor. Professor Clément.
Do you promise to work in accordance in the principle of scientific integrity at all time, to be careful and honest, transparent, independent and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confirm upon you, Marta Nazzari, the degree of doctor and grant you all the right attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other member of the committee and affixed with the official seal of the university.
Saved by the bell.
Okay, so dear Dr. Nazzari, dear Marta, the time has come for one of the last steps of your PhD defense, which is the laudatio. And I'm sure people expect me to come with what type of PhD student you were. So every PhD student is unique. So what's your story? So in your case, your story was already unique from the start, from my point of view. Because in late spring 2019, when the time has come for me to hire a PhD student for the screen project, so I wrote a job application, a job description, wanted someone that could do the lab work and the bioinformatics, which usually do not exist, but one can hope. So I sent this job description, I received 70 CV, and usually, I'm used to the process of having interview with the head of the department. So I go to Jos Klenians at the time, head of the department, and he told me, I'm afraid I won't have the time to read those 70 CVs. So I propose that you do the first round yourself, and you select the 2 or 3 best candidates, and we do a second round together. So I did that, I selected 5 candidates, and you were the very first interview, and that was my very first one-to-one interview. And I have to say, that really changed the rhythm of the interview. And you can already see, if you would be able to make a nice team and a nice connection with the candidate. So we did an interview, and I discovered a very enthusiastic and a very interesting candidate. You had a lot of questions. I think we discussed for more than an hour. You had a lot of questions. I remember 2 things that make me decide for you at the end. The first one was your motivation to become a bioinformatician. So, most bioinformaticians do big bioinformatics because they want to have fun with data, or because they hate the lab work, or both, like myself. For you, the main motivation was that you wanted to be sure to make a PhD that would not involve animal testing, and I find that a very noble motivation to become a bioinformatician. But so the real decision maker that made me select you at the end is your answer to questions I like to ask in interviews. So in interview, I like to ask these questions where I describe 2 archetypes of scientists. One being the chaotic, creative scientist who can generate a lot of ideas, but sometimes have a hard time to focus. And the very structured and organized scientist who sometimes could lack the creativity. So I like to ask that and ask the candidate to choose. And because that was my very first time asking that, I guess I did not choose my word very nicely, because I don't remember the way I said it, but I clearly remember your answer, and you told me, despite the fact that you seem to have a very bad opinion of organized and structured scientists, I'm clearly a structured and organized person. And I really like this answer because the quality I'm really looking in a PhD student is honesty, and someone that I can trust. And here, despite the fact that you were considering I wanted another answer, which was not the case, you still give your honest answer. And to me, that was really a decision breaker to select you at the end. So I came back to yours and say, I think I have 2 or 3 candidates that could be nice PhD candidates. And yours told me, well, now I'm living in summer holiday, so I won't be able to do a second round with you finally. We cannot make those students wait. So he told me, and people knowing yours recognize this sentence, he told me, you just have to pick your best candidates and live with the consequences of your choice for the next 4 years. And this is where I realize really, the responsibility which is taking a PhD student. It's a life-changing for the PhD student, of course. It has consequences on the career of the supervisor too, and it's a lot of money. Sometimes even more than initially anticipated, I have to say. But I offer you the position, and at the beginning I was mostly just the 2 of us, and then we asked Twan van der Bucken to join us because at the end you did both lab work and bioinformatics, and so those rare profiles I was looking for, you really became that these days. So you started in September, you joined with your boyfriend, Giulio, in the room. You decided along the way to settle in Eindhoven, which did not help the logistic of your PhD. And so I was, you keep surprising me for those 4 years, because you are a difficult person to read, and I told you that several times. So to give an example, at the beginning of the project, you were very, very proactive, sending emails to everyone in the consortium, organizing meetings, and I think it's very rare for first-year students to be so much engaged, and so much forward with people. So I thought you were a very extrovert, very social person, and I discovered later on that's not necessarily the case. Another funny example, maybe we had COVID, and then we were from home for a while, and then we come back, we have meetings with mask, and then you remove the mask, and I noticed that you have a nose ring, and for me that was surprising. I was not expecting that from you. So you keep like this, you are a difficult person to put in a box, which I think is a very nice compliment. And you keep, I have more surprising things that to discuss in the reception if you want. But so globally, I think I'm not sure I still understand you exactly after those 4 years, but for sure I think you have been a very good PhD student, very self-driven, and this was also recognized during the defense here by the members. You really is someone who wants to be in control of your PhD, and if I compare with other experience I had with PhD defense, with PhD, where people expect you, the supervisor, to be on the driving seat. Here you really wanted me, I believe, to be as a co-pilot instead of picking the road. You wanted me to advise on the possible road, and you would be in charge of picking the road. And I think we did a nice job. So to conclude on your future now, you want to pursue a career in companies, and I told you I will repeat that here. I think it's a pity because you really have, I think, self-driven and independent mindset, which would make would really shine in academia. I'm not sure it will be as much appreciated in company, but I'm happy to be proven wrong in the future. In any case, and to conclude with the tradition, with a few words in your own tongue, ti auguro il meglio per il tuo futuro.
Thank you. That was very good.
Esteemed Dr. Nazzari, dear Marta, it's my great pleasure to congratulate you with your doctorate, and I congratulate you also on behalf of Maastricht University. And I would like to share some impressions with you of your defense today. We have seen a very clear presentation, a very clear summary, and I hope and I think that it was also very clear to non-experts, like the people here in the audience here and also of the live stream. I guess that they will have very clear the idea of what you did in the last 4 or 5 years. Your thesis, I would say in 3 S's, was solid, systematic and sound. And you have used quite a lot of different approaches to a complex subject. And that has generated an overwhelming wealth of data. With respect to your defense, we have seen that you have answered the questions of the opponents in a quite self-confident and honest way. We appreciate that, and the committee is satisfied with your defense. And I would like to congratulate not only you, but also your 2 supervisors, Dr. Florian Clément and Dr. Twan van den Beuke. Congratulations with the results that we have seen in this today in a wonderful thesis and in a good defense. And of course, I would also like to include in my congratulations, and I hope you understand the English, your family here on the first row, your parents, other members of your family, I think your brother and your partner. Congratulations with what your family member has achieved today. Congratulations also to all your former colleagues and friends here in the audience and also following the live stream. And I would like to wish you a successful professional career and a happy personal life.
Thank you.
I would like to thank all members of both committees, both the Thesis Assessment Committee and also the supervisors, and for the fact that all of you, all members of the 2 committees, have critically read the manuscript. And Maastricht University appreciates your contributions and particularly appreciates the external members of the committee. And I would like to give the opportunity, before I close this ceremony, to one of our members who is online present. And may I invite you, Professor Lechner, to congratulate the young doctor. Please go ahead.
Thank you very much. Indeed, Dr. Nazzari, very nice to hear that. I'm sure you'll be hearing that a lot in the future. That beautiful new title you have. Congratulations to you. It was quite exciting to find a date for today's ceremony. That must have been quite stressful for you. I'm glad in the end that you were able to find a date. I wish I could be there in person to congratulate you, but allow me to congratulate you from afar. It was an excellent defense, and I enjoyed, again, reading your thesis. And the nice thing about it is that all of that data is publicly available, and it was your legacy, and it will continue to be used in the future, I'm sure. I'll finish by just referring to your beautiful proposition. It's hard to make predictions, especially about the future. But I would say, if I could make a prediction, I think your future is very bright, and I wish you all the best and much success in your future. Thank you very much.
Thank you very much. I really appreciate it.
Thank you very much, Professor Lechner. Before I close this academic ceremony, I would like to indicate that we will take a picture here with Professor Lechner still on the screen. I would like to invite the Neer family to stay here, and if after closing of the ceremony, I would like to invite the people here in this aula to go to the rafter and where we will have a reception. Hereby I close this academic ceremony at Maastricht University.
All right, please.
So, Marta, may I invite you to stand here?